Hi, my name is William Saarinen and I have nothing to declare. I'm a medical physicist from Helsinki University Hospital. In this study I help to analyze clinical susceptibility weighted images or SWIs from children suffering from a disease known as aspartylglucosaminuria or AGU. However, the methods we used in the study are general and could be applied to study other diseases that have similar behavior of iron accumulation. AGU itself is a rare lysosomal storage disorder. It causes developmental delay in childhood that leads to intellectual disability at school age and eventually to the untimely death of the patient. While there is no approved treatment available to cure AGU or even slow it down, promising clinical trials with novel medicine have emerged lately. The aim of our study was to evaluate if clinically scanned SWIs could be used as an MRI biomarker to follow the progress of this disease or even as a tool to evaluate the effectivity of these new treatments. An axial slice from a healthy subject on the left and a patient on the right visualizes the striking difference in the signal intensities. Now if we zoom in, the effect is even more prominent. Besides the magnitude SWI, clinical scan produces also a phase-filtered SWI. Our scanner manufacturer uses so-called left-handed system, so iron is observed as hyperintensity in these phase-filtered SWIs. First, we registered our magnitude SWIs to MNI 152T1 image. Second, we applied the obtained transformations to the phase-filtered SWIs. And third, we segmented the thalami using the structural atlas. We observed the group-wise differences in the whole thalamus measured from the phase-filtered SWIs. The Weyl plot suggests that the patient group has an overall higher signal. The difference between group-wise averages was deemed statistically significant with p-value of 0.01 with an effect size of 0.97. However, when we plot the average signal on the y-axis against subject's age on the x-axis, we see that iron accumulates in neither of the groups. A two-way analysis of variance agreed with this as age-related effect was not found in either of the groups. This was a bit disappointing considering that our hypothesis was that the iron would accumulate to the thalami of the patient as they grew older. Now if we recall from the previews, iron seemed to accumulate only to specific regions in the thalamus. We used a voxel-wise permutation test to locate these regions. With the voxel-wise permutation test, we successfully located the regions within patients' thalami that contained more iron compared to the healthy control group. The located regions are shown as a color map on the MNI 152T1 image. So basically, we went from comparing the whole thalamus to compare much smaller subvolumes of the thalamus. We repeated the group-wise comparisons using only the data from the subvolumes. The difference between the control and the patient group is quite evident based on the Weyl plot. The difference between the group-wise averages was deemed statistically significant with p-value less than 0.01 using a two-sample t-test with unique variances. The effect size was 3.52 evaluated using Cohen's d, indicating that difference is very large. Finally, we repeated the linear regression analysis using only the data from the subvolumes. On the y-axis is the average signal and on the x-axis is the age of the subject. For the control group, which is shown in yellow, results were quite similar as before. However, for the patient group, which is shown in purple, we now observe a strong linear correlation with R-squared of 0.65. The slope term was also deemed statistically significant using the two-way analysis of variance with p-value being less than 0.01. This confirms our hypothesis. As the patient grew older, more iron accumulates to their thalamus. Moreover, this provides a tool to evaluate the effectivity of a treatment to this or similar diseases, as we hypothesized that any working treatment should decrease or slow down the accumulation of iron, thus a treated patient group would produce a more gentle slope compared to a non-treated group. Thank you for watching this presentation. My name was William Saarinen. Have a nice ASMR.